Guys, so we just finished running our gel and now we're going to take a picture of it and we're going to cut it out. So I have fresh gloves on because I haven't touched any ethidium bromide yet, and I'm going to try to keep my right hand clean so I can touch some non-contaminated stuff and use the computer. So I'm going to come over to our gel box. So in order to do that, I'm actually just going to put my hand in my pocket. Make sure I can't touch anything. So I come over to our gel box and take off our top. See how easy that is. And the first thing you can see is our gel is just sitting in here. So I'm just going to take it out. I can reach in there. There's no electricity running through it or anything. And if you're to look at this, if I place it down there, you can see that there's two bands, that light blue and the dark blue. And that's just the loading buffer. That kind of tells you how far your gel is run. If that bottom blue band starts to get low, start to worry. If the top blue band gets really low, then you really want to start worrying. So I'm just going to grab my gel and we're going to take a quick picture of it. So again, I'm only using my left hand. We're going to put it in our imaging box and shut it. Turn on the UV light, and now with my clean right hand, I can come over and use this mouse and then just snap a quick photo. And then I'm going to turn off the UV light. I don't want to irradiate my DNA for too long, otherwise it's going to start to get messed up and degrade, and it's going to be hard to clone with. So our picture loaded, and we can open it, and now we have a nice little image of our gel. And we can see that we have our two vector bands, which is great, and then I want that part right around there. So now I'm going to show you how to cut it out. So now that I'm done using the computer, I'm going to get my right hand contaminated, but I can't touch anything else after that. So I'm going to leave this image up while my hand is still, my hand is still clean. I'm going to zoom in on it a little bit, and I can come and refer to this before I start cutting it out. I can decide, okay, I want 1, 2, 3. So I'm going to open up our gel box again. We're going to grab our gel. I'm going to throw it onto a little the top of a old petri dish. Just kind of reuse those. They make good little cutting surfaces. And we're going to come into the gel box. So gel box has two settings, 302, 365. 302 is definitely a little bit stronger. So if you're going to use this for cutting out DNA, just be aware that every time you're imaging and you're looking at it, you're killing your DNA. So to turn it on, you just click on, and then it'll turn purple. You always want to keep a face shield in between you and the light bulbs, otherwise you'll go blind real quick. So right now you can't really see anything on our gel, and you can't see anything, I'll let you get in there. You can't see anything if I just have the DNA above the gel shield, but as soon as I put it underneath, we see a little bit of DNA starts to light up. So let's see here, how are we going to fit this? I'll get in there. And I don't know, can you get a good view? Okay. So I'm just going to cut out my fragments using a knife. Now, how I cut out my fragments is I just do a little slice on the top. So I'll start off with my vectors. I give a slice, and then a slice on the bottom, and then slices vertically. Oops, and my gel's falling apart. And that makes it pretty easy. And then it's hard to see, but I have my small fragment down there. And 1, 2, and so now I have all the cuts made, and I'm just gonna try to pop them out using that using the scalpel. And then it's hard to see, but the bottom part doesn't have any DNA, so I just try to trim off as much excess agarose as I can. So I'm gonna pop these up. This is a better example. You could see that the bottom of the band is right there, and we have a bunch of excess agarose right around there. So I'm just going to chop that bottom part off. You want to try to get rid of as much agarose as possible. And then I just kind of push it back. So now we have our 3 gel fragments, and we have to remember where they are. So 1, 2, and 3. And we're done cutting. So turn off the UV when you're done. You don't want it overheating. And also, I guess kind of an additional safety precaution, you can wear eye goggles if you want, and we also have a face shield. So one of these things, each layer takes off about 99%. So 1 layer is 99%, 2 layers is 99.99%, 3 layers is 99.9999%. So protection is key. So I'm going to come over here and I'm just going to put this tray down, and now I have my 3 DNA fragments right sitting on there. I'm going to now take off my gloves because I don't want to get anything contaminated. And off those, I'll just grab another thing of gloves. And now I'm going to put my gel fragments inside of these tubes. And from here, what we've done is we've basically taken a liquid DNA and we've run it on a gel, and we've separated it by sizes, which we've already calculated. We know where to expect our sizes, and then once we image them, we can actually cut out the little bit of DNA, and we know exactly the size of DNA, which corresponds to the sequence of DNA that we've figured out on our computers. So we have these gel fragments that we know exactly what they are, and now we're going to purify them, put them back into a liquid form. We're going to go from gel to liquid. So we've started off with liquid, digested, run it on a gel in order to size it, cut it off with that gel, and now we're going to re-purify it. So I have these tubes that are pre-labeled. So we have 1, 2, 3 tubes, and the way I get my DNA, everybody has their own little technique, is I just get little pipette tips, and I stab my fragment. There it is. I don't know if you can see it, maybe against that thingy. So there's my fragment, and I just kind of pop it in that tube. So 1, here's our vector, we have 2 vectors, here's 2. So that is how you cut out gel, and we'll be back and I'll show you how to do a gel cleanup.